Hello everyone, my name is Blake and we are going to be going over the animal cell today. So, as you can see here, I have a crudely drawn animal cell. We can tell it's an animal cell here because the vacuole marked here is not large. In a plant cell it is much larger. There's the vacuole. We also can tell that there's no cell wall, there's just a cell membrane, which is marked here. It's got two layers. It's a phospholipid bilayer, which means it is not, water cannot go through this component. We also know that there's no chloroplast, which you've probably heard from biology class, is responsible for photosynthesis through a chemical called chlorophyll. But this is not about photosynthesis. This video is about a general overview of the animal cell. So, if you follow my cursor here, here is the nucleus. What do we know about the nucleus? We know that it holds the DNA. When it's condensed form, it is known as a chromosome. In its loose form, it is called chromatin. The condensed form is also called heterochromatin, and the non-condensed form is called euchromatin. So, the more you know. Next, we have the nucleolus. Here, ribosomal RNA is created, and ribosomal RNA is shipped and created. It's translated and transcribed in the nucleolus. So it can create ribosomes and then ship them off to the ER, or the endoplasmic reticulum, but we'll go into that later. So there's the nucleolus and the nucleus. So we now know that the nucleus houses all the DNA of the cell. Not all the DNA. Some DNA is actually in the mitochondria, but a majority of the DNA for the cell is held in the nucleus. The nucleolus holds the RNA for ribosomes, and we know we're going to go into ribosomes next. So ribosomes are these little speckles on top of the rough ER. They make proteins. So, if you know the central dogma of biology, you know that DNA is transcribed into RNA, and RNA is translated into protein. The translation aspect happens in ribosomes, which are right here, on the rough endoplasmic reticulum. So the rough endoplasmic reticulum is known for also making proteins that are sensitive to the cytoplasm, which is the stuff surrounding the cell organelles. So I'll just go put that right here. I'll just put cytoplasm. Nope. Yeah. So the cytoplasm environment is different from the endoplasmic reticulum. Think of it as like a membrane where the proteins are made and then housed before they're secreted in vacuoles, or not vacuoles. Endosomes is the word for them. You don't really need to know that unless you're getting into the more specific stuff. For med school, you probably need to know that in a little bit more detail, but this is going to be a very basic video, and based upon how this does, we'll see what direction we take this channel. So now that we've gone over the rough endoplasmic reticulum, which we know makes proteins, we're going to move to the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum, instead of synthesizing proteins, it synthesizes lipids, so lipid biomolecules. These could be signaling molecules, these could be just fat, like molecules that are responsible for just any lipid-based molecule and that becomes important later. But I think we're just going to go over the function of each, the bare minimum function of each of these organelles, so that our understanding we can set a foundation for our understanding of the cell. Next, the Golgi complex. So after proteins are created from the rough endoplasmic reticulum by ribosomes, they can either be shipped to the membrane, where they can go through the barrier via protein transporters or protein transport channels, transmembrane protein channels, or they can just be secreted out of the cell, depending on the protein synthesized. The Golgi complex, located here, is where post-translational modifications are made. So if we go back to the central dogma, we have DNA transcription is transcribed into RNA. RNA is translated, into proteins, and these proteins are shipped to the Golgi complex for post-translational modifications. These could be glycosylations. You could attach signaling molecules essentially to these proteins, so they have more clear instructions on where they can go after they've been translated from the RNA in the ribosomes. This is important because your cells need to be able to send these proteins that they're making to specific regions of the body, and the way they do that is the Golgi complex. They call it the packaging center. So after the package has been created, it goes to the packaging center to get labeled. So that's one way you can think about it. Next, we'll go to the mitochondria. Most of y'all know this as the powerhouse of the cell, which is mostly true, and also is responsible for apoptosis, if you know what that means, which is cell death, programmed cell death. And it happens when the membrane is punctured and releases caspases into the cytoplasm. Once caspases have been released into the cytoplasm, they signal programmed cell death or apoptosis. I know that I think it's called cytochrome C? Yeah. Well, one of the two. We don't need to know that for this video, but it definitely, once that molecule gets into the cytoplasm, the cell picks it up and initiates cell death. And this is called apoptosis. So, next, we'll go to the vacuole. The vacuole holds the moisture and the water supply of the cell. It also holds a bunch of nutrients that might be necessary for later. It just holds them there in a safe space to make sure that they don't get targeted by other cells or other organelles in the cell that might destroy them, or, because you don't want your storage getting destroyed by anything, so you just house it in that storage compartment. Phagosomes. So a lot of my work was done with lysosome activity, but phagosomes are what engulf either pathogenic or foreign substances in the cell. So after they are phagocytosed, they combine with lysosomes, which have chemicals in them that break down these substances. Some use a chemical called cysteine. You don't need to know that unless you're trying to go to med school, and then you do need to know that. and they contribute to the acidity of the phagosome. When they bind together, when these two come together, they are called a phagolysosome. Sorry about that. Phagolysosome. And here they are combined together, and after they are combined together, they destroy the substance, and depending on the cell, see, if this was an immune cell, it would preserve some of the antigens and present it to the adaptive immune system, but that's not important for this video. But that's just a little bit more in depth. The important thing is that phagosomes phagocytose, lysosomes break stuff down, and they do this when they come together as a phagolysosome. Okay, so we've already gone over a majority of this. The second to last thing we're going to do is talk about the cytoplasm. This is just a neutral. The cells, like they say, like 70% of your body is water. They're talking about the cytoplasm. It's not exactly water because it has a bunch of proteins and cytokines and other things floating around there, signaling molecules for the cell. Got ribosomes speckled in, other organelles, but that's generally most of the volume of the cell. And then we have the cell membrane. So what is the cell membrane? The cell membrane is a phospholipid bilayer that prevents any molecules that are hydrophilic from entering. So if you look at these heads here, I apologize for my really professional drawing here. These are the heads. See these circles? They're phosphate groups. And they are polar because they have a phosphate and an oxygen. If you note the structure of water and the famous saying, likes dissolve likes, you realize that H2O has an oxygen in it, and phosphate is a phosphate molecule with an oxygen, or 4 oxygens. But these are phospholipid, or phosphoheads, so they are able to interact with water and not have an adverse reaction. However, these internal parts of this, the phospholipid, or the fatty acid tails, so the phospholipid heads are on the top and the innermost part of the membrane, but the inside of the membrane, right in here, these are all fatty acid tails. So if you can imagine, like, the same hydrophobicity, they're hydrophobic, so they don't allow water to get through, which is why the inside of the membrane has a different pH or different environment as compared to the outside of the cell. It's because of those fatty acid tails. They prevent water from going through regardless. There are other ways to get inside the cell. Like I've marked here, we have there's ion channels. We have in some cells we have ATP, or sodium potassium pumps. We have transmembrane channels. You'll learn about active transport, passive transport, and facilitated transport. Active transport utilizes a molecule called ATP, and adenosine triphosphate. This is the main source of energy. Whenever you hear the word active, you should be thinking ATP. It takes energy. If you have facilitative transport, that means it's utilizing a transportation gradient, or concentration gradient, excuse me, and it's using the process of diffusion to transport things across a membrane. For water, this concentration gradient is called osmosis. In regards to other ions and molecules, it's called facilitative diffusion. And then you have passive transport, where it can just go through these channels or through the cell or cell membrane, because it's passive. It doesn't really need anything, it just goes through. Now that we've gotten all that out of the way, we're going to talk about the difference between animal cell and a plant cell. We're going to do that real quickly. So there's three main differences with a plant cell and an animal cell. I'm going to draw it right here. Again, excuse my artistic genius. Oh goodness. Alright, hold on. Excuse my artistic geniusness. I know, it should be in a museum or an art gallery. But that is the cell wall. In the cell wall, you have sugars that form a barrier. And these sugars are called cellulose. Cellulose has beta 1,4 linkages, I believe, and they're not digestible. So they form a nice barrier, an extra layer of protection. Additionally, so in addition to the cell wall, the vacuole is going to be bigger. It's going to be like much larger, so it'll be like that, which also helps prop up the plant cell, because plants need a little bit more water. We need water, but we don't need water like plants need water. Plants really need water. That's why they have a bigger vacuole. And then you have chloroplasts. And inside these chloroplasts, you're going to have these discs. These discs absorb sunlight and conduct photosynthesis through a molecule called chlorophyll. Chloroplast. And inside the chloroplast, you're going to have chlorophyll. And chlorophyll basically takes in sunlight and carbon dioxide and water and makes sugar, glucose, and oxygen. I can write out the formula, but honestly I'll save that for another video. So this is the basic dogma of cells. I hope you found this useful, and let me know what I should do in the next video. Thank you.